Hi, Dr. Hurt, or whoever else is watching this right now. My study is called The Effects of Artificial Protection Structures on Soft Substrate Intertidal Zone Communities in the Chesapeake Bay at Horn Point Laboratory. So it's a kind of wordy title, but it's a pretty picture. So this study is to observe the reaction of different organisms in the intertidal zone of the Chesapeake Bay by Horn Point Laboratory when they are presented with an artificial structure. So an experiment like this was done in 2007 by Bertassi, I think that's how it's pronounced, and a bunch of other scientists. What they did was they observed low-crested structures in the North Adriatic Sea, and that ended up being a really good way for them to study the organisms in the area and the diversity. So hopefully that kind of validates the way that I set up this experiment. I've kind of tried to model it after that, and hopefully we can really, I would have been able to really study the organisms in the area. And another cool thing that I read about when I was researching for this is coastal defense structures, which are starting to be implemented. What they do is they protect coastal areas from erosion and from global flooding, because those areas are really important in country development and also for the ecosystems. And before coastal defense structures can really be set up in different places, that place needs to be studied to see if a structure, an artificial structure there, will really hurt the environment, if it'll change anything. And so I thought that this could be a kind of experiment to give an idea of the way that might change the environment, how it would affect it, how the organisms would adapt to it or use it. Okay, materials and methods. So Horn Point Laboratory has access to a beach. I don't know if it's a sandy beach or a rocky beach. I'm guessing it's sandy, but the picture I have there is rocky, so I'm not sure. I've never been. But it also has access to a salt marsh, which I'll use too. So there's 2 concrete masonry units, which are basically just cinder blocks. And one would have been put in the benthic zone of the beach area, in the intertidal zone. Another one would have been put in the salt marsh. And 12 hours they will be surveyed, which makes 4 surveys of each unit. And what I'll do is I'll have a chart, and on that chart the amount of organisms that are there will be documented, and the species of those organisms will be documented, and their size, any unusual or unique behaviors, the congregation, how close together they are, if they're spread apart, if they're clustered. And with every organism that is documented on the chart, I will also have some sort of visual record of them with my GoPro camera. So that's a picture of the GoPro I would have been using. Those photos and videos will be recorded, hopefully in the same order that I put them on the chart, so it'll be really easy to keep track of. And that way I'll just be able to have a picture with everything, with all my data, and I thought that would just be a cool thing to add. So normally on artificial structure, you would expect some algae to grow, especially because of eutrophication in the Chesapeake Bay. A lot of fertilizers causing a lot more algal blooms. But 48 hours will not be enough for any algae to grow on the unit. So sessile organisms, though, will most likely attach themselves, anchor down to it. So we expect things like periwinkle snails or tunicates, maybe some bivalves, oysters, things like that. Maybe some small crustaceans, small crabs, crawfish, crawdads, whatever they're called. And since a lot of species of fish tend to use estuaries as a sort of nursery, then there might be some juvenile fish around the structure. It would be pretty lucky if we could see them, if they wouldn't, before they swim away when we go to survey, but I think that would be really cool if we could see some of those and hopefully try to identify them. Okay, so why does a study like this matter? I talked about that a little bit in the introduction, but I thought a really important thing was that it could give some sort of insight to the ecological health of this area. So I talked about how it's expected to see periwinkle snails, but if no periwinkle snails show up on the block within those 48 hours, then that could be some evidence that maybe periwinkle snails aren't doing so great in the moment, or it could also just be something with some other factor that I don't know. But I just thought that could apply. Also, when Bertazzi and all the other scientists did their experiment in 2007, they saw a lot of diversity. And what they did was they compared that with the diversity seen by other similar studies in the area by Parisi in 1985 and Ambrogi in 1990. And what they saw was that in 1985 there was not very much diversity at all. And in 1990 there was a little bit more. And in 2007 they saw a lot, and that was good news because they had been going to a lot of efforts to help the environment in that area from pollution. So that just was very encouraging results to show a lot of increase in diversity. And since there's not really any studies that I could find of diversity in the same Horn Point Laboratory area from before, I thought that the results of my study could serve as a kind of baseline for this. So if any studies are done in the future in the same sort of way, then they could compare the diversity that they find to what I found, or whoever does this will find, and just see if diversity is increased or decreased or any of that. So yeah, I think it could have been a pretty cool experiment. Wish I could have done it, but it's okay. These are all the references that I used. Yeah, not all of them I talked about in the presentation, but a lot of the information that I got was from these. And yeah, thanks for watching, like and subscribe. Just kidding.